All right, so next we're going to focus on the neuron, which is the signal-sending cell of the nervous system. And this cell will actually be the main focus of the discussion of the nervous system that we have in Chapter 11. All right, so what are neurons? What are the characteristics and how do they send signals? That's what we'll cover in this video here. All right, so neurons, we describe them kind of three ways. We describe them as excitable cells that send signals and respond to stimuli, right? So remember that an cell that's excitable undergoes a brief change in membrane voltage in response to a particular stimulus. All right. Neurons are highly specialized for sending and conveying information in the central nervous system over long distances, and we'll see the structures that come into play to receive and send those signals long distances. And these cells are very, very highly specialized to the point that they're not really good at much else. So once you get this specialized as a cell, it's difficult for you to do other things like undergo mitosis. So these cells are so highly specialized that they've lost the ability to undergo mitosis, which makes sense, right? Because we know that certain types of damage to the brain and to the spinal cord never heal, right? Because these cells are constantly sending signals back and forth and receiving information in our body and sending information to different parts of our body. These are extremely active cells in that nervous system tissue and neurons in general require pretty much constant oxygen and blood delivery to them, otherwise they die within just a few minutes. All right, so we kind of discussed a little bit about general characteristics of neurons. We'll look at specific characteristics and structures associated with neurons. All right. So our neuron, like any other cell, has a cell body, or the main part of the cell, which we find most of the organelles. In this case, the cell body is also referred to as the soma in the neurons, and neurons, or neuron somas, or neuron cell bodies, are not really all that different from other cells, and that they contain sort of the typical complement of organelles that we're used to seeing in cells. So remember that cells have to have a nucleus which coordinates the activity of the cell. Neurons, although they're very long cells, they only have a single nucleus per cell. They have to have a way to make ATP energy. They have to have a way to make energy for themselves, and they do so with mitochondria. And you can see some of the mitochondria in this diagram here as these little sort of red bean-shaped structures here. All right. The mitochondria make energy. The endoplasmic reticulum, recall that's a structure that surrounds the nucleus, and you can kind of see it as these flattened tunnels that are surrounding our nucleus here, and those are that's the rough endoplasmic reticulum there that helps to make proteins. And we also have the Golgi apparatus, which is in here somewhere. It's not really very specific as to which structure it is within this diagram here, but the soma contains the Golgi apparatus, which, recall, that receives material and proteins from the endoplasmic reticulum and shuttles and moves them to different parts of the cell. And I mentioned already that the neuron has a single nucleus per cell, even though it's a very, very long cell. One structure that I wanted to show, and they give a little bit different name to, they give a slightly different name to, is part of the endoplasmic reticulum. Recall that it's the rough endoplasmic reticulum that makes things like proteins. In neurons, they call these a little bit different structure. They have a slightly different name for this structure. It's called the Nissl bodies. In this diagram here, what you're seeing, this triangular structure here, is the soma of the neuron. The nucleus is within this little structure here. Or sorry, I believe that's the nucleolus, actually. I think the highlighted portion here is actually the nucleus. Yes, it is. It looks like it's N is referring to nucleus here. And you can see that these little sort of dark spots that are surrounding the nucleus, those are actually the kind of clustered appearance, or the endoplasmic reticulum has kind of a clustered appearance about it, rather than one continuous appearance. The reason why the Nissl bodies, or the endoplasmic reticulum, looks clustered in the way that it is because the cytoskeleton of the neuron, which in this case is called a neurofibril, They essentially kind of thread their way through the soma, and what happens is that because of the way that the neurofibrils thread their way through the soma, the Nissl bodies, or the rough endoplasmic reticulum, kind of takes on sort of a scattered appearance, and it appears as little little orbs surrounding the nucleus here. Okay, but functionally it's the same. The Nissl bodies are the exact same structure as the rough endoplasmic reticulum. All right. The nervous system tends to cluster cells together that have similar functions associated with them. So in certain places, in certain parts of the nervous system, both central and peripheral nervous system, we find that there are clusters of cell bodies, clusters of neurons together that go to similar, and these neurons communicate with a particular muscle or a particular gland, and the somas of all those neurons just happen to be clustered or gathered together in one location. So if we encounter a structure or a cluster of neuron cell bodies that is located within the central nervous system, that is within the brain of the spinal cord, we call that a nucleus, right? And so be careful here with this term because the term nucleus in context of the central nervous system is spelled exactly the same way as the nucleus of the cell, right? So these are both structures that are spelled the same way, but the context of which we use the information is a little bit different. So a cluster of neuron cell bodies in the central nervous system is a nucleus, whereas the nucleus of the cell is the control center of our cell. All right, so we saw that in the central nervous system there are clusters of cell bodies. The peripheral nervous system also has localized clusters of cell bodies. And in this case, if they're located in the peripheral nervous system, they tend to appear as these sort of little knot-like swellings in the nerves, and we call these sort of knot-like swellings, we call them ganglia, which is plural. Singular, that would be a ganglion. So here's a diagram here of the spinal cord with nerves emerging from it, and you can see these sort of enlarged regions of the nerve here. Those are what we call the spinal ganglia, and they're also shown in this diagram here. Okay? So as I mentioned, they always appear as these knot-like swellings in nerves. So those are localized clusters of cell bodies. All right. So extending away from the cell body, and in this diagram here, there are several that extend away from the cell body, are processes that are called dendrites. Okay, so these are branch-like processes that always extend away from the soma. And the term dendro here, you kind of think of as like, so it makes reference to a tree, and really these structures are named because of their resemblance to tree branches. Okay. The dendrites. What do the dendrites do? The dendrites are receptive to the stimuli. That is, they're receptive to the changes in the environment, and they actually create an electrical signal that's going to spread a little ways down the dendrite and into the soma of the cell. So the types of stimuli that they're receptive to stimuli from other neurons. That is, another neuron can release a neurotransmitter and signal the dendrite of a cell. Other cells can communicate with dendrites here. And then environmental stimuli can also stimulate dendrites to begin sending signals toward their cell body. And the type of signal that the dendrite creates, it's an electrical signal, but it's not a long distance electrical signal. It's only a very short distance electrical signal, and we call this electrical signal a graded potential. And we'll talk a little bit later. I've got another video that highlights what a graded potential is and what kind of signal that is, what kind of information it carries, things like that. But this electrical signal travels along the plasma membrane of our dendrites, always toward our cell body or towards our soma. All right, so we've seen the dendrites, and all neurons, no matter what kind of neuron it is, neurons usually are quite variable in how many dendrites they have, but neurons are always the same in that they have a single, typically very long process that extends away from the soma called the axon. The axon, it's typically not a very long structure, but in some neurons it's extremely long. That is, in some neurons whose cell bodies are in our spinal cord, and they send axons down to make synapses with the muscles in our feet that control the movement of our feet and our toes. These axons can extend all the way from the spinal cord down through the leg and to those muscles of the foot. So for that reason, some axons can be as long as 3 feet. Like I said, most are typically very short, but there are some exceptions where these structures are very, very long. All right. So just like cell bodies are clustered together, the nervous system also likes to group or cluster together axons, especially for axons that travel to one particular location in the body or make a connection with a particular muscle or a gland. Okay, so axons also form cluster groups in certain regions of the nervous system. If this clustered group of axons, or if this bundle of axons is located in the central nervous system, we call it a tract. Right? If this bundle or cluster of axons is located in the peripheral nervous system, we call it a nerve. Right? So a nerve is really nothing more than a large group of axons that's bundled together. All right. And our axon has to connect to our soma, and it connects to the soma using this cone-shaped process called the axon hillock. And we'll see the axon hillock will be an important structure later on for the long-distance signaling of the action potential that we'll see in neurons later on. All right. All neurons have only one axon, but that doesn't mean that this axon can't branch extensively at its very end. So we've got another example of neuron here with a long axon leaving its soma or its cell body. But as the end of the axon, or as we reach the end of the axon, this axon branches many, many different times, or multiple times, so that it can make synapses or make connections with lots of other cells. And we call these branches terminal branches. So functionally, this is still one axon. It just happens to branch out at its very end, so it can make connections with lots of other cells. All right. So at the very end of the axon, so this may seem familiar. This will be kind of a little bit of a review from skeletal muscle physiology, neuromuscular junction stuff. We saw that the end of an axon expands a little bit, so it has kind of a flattened sort of bulb-shaped end that we called the axon terminal, right? And it's in this axon terminal that we have the storage of our chemical messengers, which are called neurotransmitters. And it's the axons that form junctions, or what are called synapses, with other cells, right? So the kinds of synapses that we're going to see, or we have already seen in the nervous system, we'll see that axons can make synapses with the soma or the dendrites of another neuron, or other neurons, I should say. And back in chapter 9, we saw that the axon terminal made a synapse with the skeletal muscle cell that we call the neuromuscular junction. Right, so it's within these axons where the chemical signals of the nervous system, called the neurotransmitters, are stored and released, so that and they're released from those chemical messengers are released from our axon terminal, so that they can signal our other cell, or the cell that the axon is making a synapse with. Right, and it's within these structures here, these little what I refer to as bubbles of plasma membrane that are called synaptic vesicles that store these neurotransmitters until they're released. All right, so we've seen the axon terminal. As I mentioned, the axon can be a very, very long structure. That is, some axons that connect the spinal cord to the skeletal muscles that control the movement of our toes and our foot. The axon that carries the information from our spinal cord to that muscle can be as long as 3 feet. Because of the length of this particular cell, our axon needs a very, very rapid way to communicate that information across that relatively long distance, because 3 feet is a very long distance for a cell. So we need a very fast, high-speed way of sending signals down those axons. And so axons create and send these electrical signals that we call action potentials. So these are the long-distance, fast electrical signals that are going to help our neurons communicate with other cells outside of the brain or the spinal cord. All right. Action potentials, some of the fastest action potentials can travel along the axon at speeds of around 300 miles an hour. It's quite fast. So essentially think almost instantaneous arrival of a signal at a muscle or a gland with action potentials that are that speed. All right. Action potentials always travel away from our soma, away from our cell body, and down towards our axon terminals here. So this little arrow here is indicating the direction of action potential movement. Again, always towards the axon terminals, away from the cell body. All right. As we kind of start to reach the end of our tutorial, our overview of neurons here, I wanted to kind of finish up the discussion of axons with talking about movement of materials along axons, because axons, because of their length, they don't make the materials that they need. They don't have the organelles and the structures to make proteins, to make plasma membranes, to make other cellular materials that they need. It's the soma is really where all of the synthesis of proteins and plasma membranes and things like that are occurring, because it has the nucleus, it has the rough endoplasmic reticulum. So axons, material has to be shuttled up and down within the axon, or within the interior of the axon. And so any kind of material that's moving down the axon, that is any proteins that are being made, synaptic vesicles, neurotransmitter that's being made, has to be moved or shuttled inside of the axon down to the very end, down to the very end of the axon terminals. And anytime we have movement of material away from the cell body or away from the soma and toward the axon terminals, we call that anterograde movement. Whereas material like damaged organelles, proteins that need to be broken down, things like that, cellular wastes that need to be to be eliminated, tend to move from the axon terminals up towards the soma, towards the nucleus. And anytime we have movement up the axon, away from the axon terminals and towards the cell body, we call retrograde movement. All right. So we've looked at general characteristics of neurons right now. I wanted to finish this up with talking about the three different types of neurons and their functions in the nervous system. And there are three types of neurons that differ in not only their shape, they differ in the number of dendrites, that is the number of processes that extend away from the soma. They also differ in the type or of information that they're going to convey or carry in the nervous system. That is, some neurons only convey sensory information, some are strictly integration neurons, and some are carrying motor commands away from the central nervous system toward the muscles or glands. All right. And these three types of neurons that we're going to overview just briefly here. The first type is called a multipolar neuron. Second is called a bipolar neuron. And our third type is called a unipolar neuron. All right, so we'll look at structure and function of multipolar neurons first. Right. So the handout that I gave you on Moodle is an example of a multipolar neuron. And so multipolar neurons are typically, from the cell body, they have a number of dendrites that extend away from them. So these dendrites can often be, there can often be a few thousand dendrites that extend away from a cell body. And then a very long axon that, of course, always proceeds away from the cell body as well. All right. Multipolar neurons I bring up first because these are the most common of the neurons in the central nervous, in the nervous system in general. So most, and that is about 99% of any neuron in the nervous system is going to be a multipolar neuron. So these are the most common. And these guys are typically associated with the central nervous system, that is, their cell bodies or their somas are typically housed in the central nervous system, but that doesn't mean that their axons can't leave the central nervous system. And indeed, we do see a lot of axons of multipolar neurons that leave the central nervous system. All right, as I mentioned, they have many dendrites, sometimes a few thousand dendrites that extend away from their cell body, and of course a single axon, as all neurons only have one axon. So what functions do we see multipolar neurons fulfilling in the nervous system? Multipolar neurons are always either integration neurons, so they always fulfill function 2, or they're motor neurons, they fulfill function 3. Right. So as integration neurons, recall that integration is the decision-making process of the central nervous system, where the central nervous system receives sensory information and then it decides how should I respond to that sensory information. Right. So integration neurons are kind of functionally the link between the sensory and motor pathways of the nervous system. Right? And recall that I said that integration is the decision-making. How should the nervous system respond to a particular stimulus in its environment? Multipolar neurons also function as motor neurons. That is, they're carrying this decision, or they're carrying information away from the central nervous system to respond to a particular stimulus that our body has received. And typically motor neurons synapse with muscle cells, that is skeletal, cardiac, or smooth muscle cells, or glands. All right, so those are the multipolar neurons. We'll turn our attention to bipolar neurons for a second. These neurons, they are relatively rare in the body in that these are sensory neurons that are only found associated with what we call the special sensory organs. And special sensory organs are things like the eyes, the ears, the nose that are carrying. The eyes convey information about light that reaches our eyes to our brain. The ears are carrying information about sound to our brain and to our central nervous system. The nose carries what's called olfactory information or chemical information of smell to our brain. Right. Bipolar neurons are named because they essentially have two processes that extend from a central cell body. They have a dendrite that can branch at its end, and these branches tend to fuse together into one single dendrite as they approach the cell body. So it does have a single dendrite, but that doesn't mean that this dendrite can't branch extensively at its endpoints. And of course, a single long axon that will extend away from the cell body. All right, so what do bipolar neurons, what function or what role do they fulfill? I mentioned that they're only associated with special sensory organs or systems, and so it makes sense that the function of bipolar neurons is strictly going to be carrying sensory information. So these guys these particular neurons are always going to be sensory neurons. Okay? They convey special sensory information from the eyes, so light, which is visual information, ears, which is auditory information, and smell, which is what we call olfactory information, to the brain. Because these neurons are always external to the brain, they are considered part of the peripheral nervous system. All right, and last but not least, we have our look at unipolar neurons. These are actually kind of an interesting neuron in terms of their shape and structure, but in general, they are also delivering sensory information to our central nervous system, but it's a different type of sensory information. It's what we call general sensory information. So this is things like touch, temperature on our skin, so hot and cold, pressure, pain from damaged tissues or chemicals or things like that. And these, as I mentioned, have kind of an interesting structure in that what they really have is this single long axon that makes up sort of most of the cell. The cell body kind of sticks off of the middle of the axon, a little stalk of tissue here. And then they have these structures at one end that are called the receptive endings, which are functionally these are dendrites. It's just that the book doesn't refer to them as dendrites because the book really uses a kind of an antiquated and old definition of dendrites, where dendrites always connect directly to the soma. In this case, the book refers to these as receptive endings because even though, as I mentioned, functionally these guys are dendrites, or functionally they behave as dendrites, they don't connect directly to the soma, they connect directly to the axon instead. So for that reason, these guys are called receptive endings and not dendrites. And at this end here, these would be the axon terminals as well. But again, lots of dendrites, but a single axon. All right. What is the function of unipolar neurons? What role do they fulfill in the nervous system? They carry general sensory information, so it's no surprise, again, that these these neurons are also always going to be sensory neurons. Right? So these guys are going to carry general sensory information like touch, temperature, pressure, and pain to the brain, to the spinal cord. Because these structures are always external to the brain or to the spinal cord, these are peripheral nervous system neurons. Okay. And as I mentioned, they're delivering general sensory information to our brain and to our spinal cord.